Our next speaker is Dong-Hua Chen from Roger Kornberg's lab. Dong-Hua is an expert in structural biology and single particle cryo-EM, and he's going to be talking about how Marlowe has helped with speed up some of the processing for the cryo-EM single particles simulations, analysis, rather.
Thank you for organizing, giving me this great opportunity. My name is Dong-Hua Chen from Professor Kornberg's lab in the Department of Structural Biology. My topic is Marlowe speed up data processing of single particle cryo-EM for large and flexible macromolecules. So nowadays, cryo-EM is a very popular tool to determine the three-dimensional structure of molecules. Cryo-EM include single particle cryo-EM and cryo-electron tomography, but here only talking about single particle cryo-EM. So this is the pipeline for single particle cryo-EM. First, purify the sample and then apply the sample onto the EM grids to prepare the cryo-EM grids. Then transfer the cryo-EM grids under liquid nitrogen temperature to the electron microscope, and then you collect your data. Depends on your project requirements. You might collect hundreds to tens of thousands images. From the images, box of the particle, then use some packages like Reliant, CryoSpark, or EM2 or other packages to process the data. And then you will get a three-dimensional structure. Depending on the resolution you get, you may be able to model your protein, and then finally deposit the model and the maps into PDB and EMDB respectively. A few years ago, we published a paper in Molecular Cell for the mediator structure. In this paper, we used the single particle cryo-EM to determine the mediator structure at a near atomic resolution. This is the cryo-EM structure for the mediator at 4.2 angstroms. We used about close to 1 million particles for this structure. The 4.2 angstrom resolution is the overall resolution, and estimated based on the showing on the left, based on the gold standard Fourier shell correlation at the 0.143 criteria. This is commonly used in the cryo-EM field. This map showing you this tail module on the bottom and head and middle module in the middle and hook on the top. And you compare to the head middle module, you can see that the tail and the hook is still very noisy. So actually, we also did the 3D variability analysis for this mediator, and this is the short movie. You can see that the tail is moving a lot, right? So the tail is flexible. In order to solve this mediator tail structure, so first we made a mask around the tail on the bottom, and then do the particle subtraction. So basically remove all the signals except the tail, and then have a new data set. And then we did this so-called focus 3D classification, sort out that subtracted data set to 10 classes. So here each class is a central slice through the density map. So among these 10 classes, you can see that only 2 of them are good. So then combine these 2 good classes, generate a new map. So this resolution is about 3.4 angstroms based on the GFFSC on the left. So this map is color-coded according to the scheme on the right. Different resolution represented, different color represented different resolution. So this example showed that the combination of particle subtraction and Fogg's 3D classification can solve the structure at the near atomic resolution. So in Compact Lab, we are trying to understand how the translation is regulated, and we are using single particle Cryo-EM to understand how these transcription components interact with each other. But there are some challenges for these single particle Cryo-EM projects. So first, this transcription complexity is huge compared to mediator. Mediator basically is about 1 megadalton. So for some complexity we are working on, have about 7 to 8 megadalton. So it's very big. So second, so with each this complex we are working on, there are many components actually are very flexible compared to even more flexible than the mediator tail I just showed you. So basically we have to collect a very, very large data set so that eventually we will have some particles left to solve the flexible domain to near atomic resolution by the focused 3D classification. So this is one of the Cryo-EM image for one of the complexes we are working on, collected from microscope at Stanford SLAC Cryo-EM Center, also called S2C2. From this 4K by 5K image, you can see quite a few particles. So each particle is about 30 nanometer times 6 nanometer in size. So for this project, we have about 5 million particles with the box size 640 by 640. So this shows you the single particle Cryo-EM data processing workflow we are using. There are many steps here, but here basically I use Relay and CryoSpark to process the data. So among these steps, these two are very GPU consuming. One is the 3D order file to reconstruct the three-dimensional map from millions particles. And then the second one is the focused 3D classification for the flexible domain. So one of the projects I mentioned have about 5 million particles with the box size 640 by 640. So we have a local workstation with 4 GPU, A6000. This Relay 3D order defined job spent about 1,000 hours, so it was 6 weeks. And I run similar job on Marlowe. I use 64 GPUs at a time, and then basically the job will get done in 2 days. So this dramatically speed up of data processing, and we can spend more time to interpret our density maps. So in summary, improve the 3.4 ancillary structures for the mediator tail determined by single particle Cryo-EM, and use the particle subtraction and focused 3D classification. We can solve the flexible domain to near atomic resolution. And last, Marlowe speed up of data processing. And I want to thank my boss, Roger Komberg, and my colleagues in the lab. And also want to thank Stanford Marlowe team. Thank you.
Where is the Cryo-EM actually at?
Cryo-EM, so right now we can solve the rigid molecule to atomic resolution. So, but here I just show the flexible molecules. So if you have very rigid molecules, you can solve the structure to 1 point something angstroms. So lots of pharmaceutical companies also trying to establish the Cryo-EM facilities, so they can use that for the medicine molecules.
Thank you.
Thank you.